So here we are, AP Biology, Chapter 16. This is kind of the start of the origin of life. And we're going to be focusing on the evolution of organisms and how one has led to the evolution, or through evolution, we have developed the diversity that we currently have today. So we're going to start with the earliest life forms in Chapter 16, and then as we move through the chapters, we'll get more and more complex. Now, when we talk about the origin and kind of the evolution of life, what we have to realize is that we're looking at or starting about when did life come from? And it came from the earliest atmosphere. And the earliest atmosphere contained things like water, carbon monoxide, carbon dioxide, nitrogen, and then maybe some methane gas. But there was definitely no oxygen, or if there was oxygen, it was in very, very small quantities. And one of the things that happens is that because of the way the environment was and because of volcanoes and lightning, we were able to actually take these non-living compounds and turn them into a living organism. And so it turns out that one of the kind of leading ideas that's out there is that bacteria is what actually changed the world. Bacteria is what actually allowed our environment to change. And it's because they have found fossil evidence of a prokaryotic organism called stromatolites. And what stromatolites are aerobic bacteria. And they are aerobic, which means that they use oxygen, and they were also photosynthetic, which allowed them to produce oxygen. And because this bacteria was producing oxygen, our environment contained oxygen. So one of the things that we're going to be looking at, or one of the things that we have looked at, are cladograms, those branching diagrams of all the diversity of life. And one of the things that everybody agrees on is that bacteria is, or the descendant, is our common ancestor. And so the common ancestor has led to plants, and it's led to insects, and it led to like dinosaurs and mammals, and then ultimately to humans. And so if we were to track through time, what we would find is that there is some major events that have played a key role in the history of life. And we'll talk about those key events as we talk about the evolution of certain organisms. And so one of the things that used to be around was the idea of spontaneous generation. And I know that in our earliest chapter, we discussed Louis Pasteur and Francis Redi, and how they did experiments that basically proved that life does not spontaneously generate, and instead life comes from other life. And the theory is that the cell theory says that cells all come from originating or pre-existing cells, which then leads to the question, well, where did cells come from? Well, this goes back to the idea of our earliest environment. And so when we look at Louis Pasteur and Francisco Redi, we're talking about them as a way of saying that, look, they proved that life does not spontaneously generate, but they also weren't able to explain how did life originate. And that experiment, or that credit, goes to a man named Stanley Miller. Stanley Miller was the guy who showed that these non-living organic compounds, when they were in a certain environment, could actually form the compounds that are found in all living organisms. And what he did was he took all the gases that were existing in the earliest atmosphere and he placed them in this apparatus, and then he subjected this apparatus to the early environment, high UV levels, lots of volcanic activity, and lightning, electricity. And what happens is that when he did this simulation, he was able to generate, okay, so here's hot water, or the ocean water boiling, collecting the water vapor. It's traveling through the atmosphere, which contains those non-living organic compounds. We're adding electricity, which would be equivalent to like lightning. And when all of those molecules condensed and cooled, what he was able to collect down here in his jar. So it ultimately would have collected down into the aquatic environments of the time were those compounds that make up things like amino acids, lipids, DNA, RNA. And so these are those macromolecules that make up living things, but they in themselves are not a living thing. And so what we were making is these early polymers. Now, how did the polymers form? Well, it turns out that if you take a hot rock or a rock and you subject it to enough UV radiation, enough lightning, they'll become very hot. And when those molecules become hot, they move around a lot, and they're possibly going to bump into each other and form a bond. Now, one of the idea is that if they were to go around bloop, and bump into each other and they were to bond, so for example, if amino acids were to bump into other amino acids, now you're starting to make a polymer chain. The problem with this is that the same energy that was making these polymers would probably also be denaturing them as well. And so what actually might have happened is one big oops or mistake. And so one of the major ideas that's out there right now is that the earliest genetic material, so RNA or DNA, actually was probably RNA. And the reason why they think this might have happened is that, again, if you take these monomers and you heat them up and they bump into each other and they form a bond, what could have happened is we could have formed an RNA molecule. RNA molecules are single-stranded, they're polymers, but remember, RNA is that intermediate molecule that we use to make proteins or we use to make other components that add to or help sustain life. So by accidentally making RNA, we now have come one step closer to creating the central dogma, so DNA to RNA to protein, that sustains all of life. And so it turns out that if we look at this, we can actually start to see how this might have been the start, but not yet, but kind of the earliest step towards making a cell. And what they have found is that RNA, even today in experiments, RNA can act as a template for its own polypeptide or own proteins. And so RNA can self-replicate more RNA because of base matching or base pairing rules. Or RNA can actually be used to form a protein or polypeptide chain. And as we know, that proteins can act as enzymes, which then make more reactions happen. And so by having our big oops formation of a polymer that happens to be RNA, we now have the basis for which to make a double-stranded RNA, which might look a little bit like DNA, and we have the foundation for which to make all of our proteins. Now, going one step further, okay, we have RNA, we might have a DNA, maybe we have protein. The first cells may not actually have been cells, but instead they might have been a co-op. And by that I meant they might have been a bunch of these polymers kind of hanging out together, trying to kind of feed off one another as like a bunch of friends working together to complete a homework assignment. Now, what they think would have happened is that by covering them in a membrane and putting all of these components inside, what we did is provide a protected environment for which the cell to operate. And so now we're starting to look a lot like a cell, right? RNA, protein, cell membrane, it's all there. So what is this earliest cell? Well, as of right now, the evidence says that the earliest cell is prokaryotic. And prokaryotic are those cells that are much larger than a virus, but they're extremely small compared to a eukaryotic cell. And as we have talked about or have mentioned before, prokaryotic cells come in a variety of shapes, sizes, colors, functions, things like that. Some of the big ones that you have to memorize is the names. So prokaryotics can be shaped like a sphere, which is like a basketball. And that is, they go by cocci, or they can be a rod, so like a hot dog shape, and we call those bacilli, or they can be spiraled, like spirochetes. Now, one of the big things about prokaryotic cells is that they're simple. They don't have a nucleus, they don't have membrane-bound organelles, yet they're a fully functioning cell. And one of the things about being a fully functioning cell is you need to be able to go out and find food. You need to be able to go out and find a good environment. And how do they do that? Well, it turns out that most prokaryotic cells will have some sort of flagella. And the flagella is nothing more than an extension of its membrane. And by allowing it to rotate, it allows for movement or locomotion. And so they were able to get to more environments. More environments means diversity, higher chance of survival. So here's some more examples of the prokaryotic flagella. You can see them in the microscope slides. And then some of them actually want to be able to stick. So they're like, wow, I really like this environment. I think I want to hang out here. How do they do that? They're going to do that with a modified flagella that's called a pili. And here's an example. You can see that there's lots of pili all around this, which allows it to stick really well to its environment that it likes. Now, what happens if a bacteria gets into an environment it doesn't like? Well, what's it going to do? It's going to make an endospore. Now, only certain bacteria have the ability to form an endospore, but basically here's what it does. It goes, oh, this environment is really stinky. It's kind of sucky. I don't want to be here. it forms this endospore, and it almost kind of goes into like hibernation, if you will. And it allows it to survive these really extreme environments until the environment gets good. It ditches the endospore, and boom, it starts reproducing like crazy. Another example in the bacteria is that there's different groups or different types of bacteria. And so we're going to start talking about all those different types. So we're going to talk about proteobacteria. We're going to talk about chlamydias, spirochetes. We're going to talk about bacteria that's gram-positive, things like cyanobacteria, which do photosynthesis. All of these things are things we're going to discuss as we kind of go through the evolution, because it all started with bacteria. But we're going to focus on all of the bacteria types and their functions in our next video.